Good morning, students. Now coming to structure of dicot seed and structure of monocot seed. Students, after the completion of fertilization, ovary it develops into a fruit. Ovary develops into a fruit. So fruit is fertilized ovary, that is post fertilization product and a product of sexual reproduction. Similarly, ovaries present inside the ovary. After fertilization, these ovaries they develop into the seeds. They develop into the seeds. So ovary is the fertilized. Fruit is the fertilized ovary and the seed is the fertilized ovule. So, they are the products of sexual reproduction. At the same time, in case of dicotyledonous seed, every seed it is represented by seed coat. Every seed is represented by seed coat. Externally, seed is covered by seed coat, and this seed coat is also differentiated into outer seed coat and inner seed coat. Outer and inner seed coat. So, this outer seed coat, this is also called testa. This is called testa. Outer seed coat is called testa. Okay, actually this testa it is developed from outer integument of the ovule. Every ovule is represented by either one or two integuments. They are the coverings of new cells. So after completion of fertilization, outer seed coat is developed from testa. Okay, this is outer seed coat. This is also called testa. It is developed from outer integument. Outer integument. This is developed from outer integument. Similarly, this is also represented by outer seed coat is also followed by inner seed coat. This inner seed coat, this inner seed coat, this is also called tegmen. This is called tegmen. Inner seed coat is called tegmen. Outer seed coat is called testa. Similarly, this inner seed coat is developed from inner integument. Inner integument. This is inner integument. In this way, after the completion of fertilization, complete ovule develops into a seed. Outer seed coat of outer integument of ovule develops into outer seed coat testa. Similarly, inner integument of ovule develops into inner seed coat, that is called tegmen, and is nothing but inner integument. For example, this is ovule, okay, and this is folliculus. This is folliculus, okay. So the part of the folliculus which is attached with the body of ovule, this is called hilum. This is hilum. Okay, what is hilum? Hilum it represents the junction between body of ovule and folliculus. The part of the folliculus which is attached with the body of ovule, that is called hilum. And after fertilization, this hilum it develops into the seed scar. Hilum develops into seed scar. Seed scar. Okay, seed scar. Hilum it develops into the seed scar, by which the seed is attached with the ovary. So, ovary is attached with the ovary by hilum. After fertilization, once this ovule develops into a seed, hilum it develops into the seed scar. This seed scar it attaches the seed with the fruit. In this way, scar is nothing but hilum. This hilum it develops into the seed scar, which helps in attachment of the seed with the fruit. Similarly, within the seed pod, in any dicotyledonous plant, in any dicotyledonous seed, within the seed pod there is a embryonal axis. Okay, embryonal axis. Embryo is there. Within the seed pod, embryo is there. Now, this embryo is also represented by two parts, that is embryonal axis. Embryonal axis and cotyledons. Two cotyledons. Embryonal axis and two cotyledons. That is the embryo present within the seed coat of dicotyledonous seed. Students, you know the structure of dicotyledonous seed is dicotyledonous embryo in previous class. What about the embryo in dicotyledonous seeds? The seed of dicot is represented by embryo. This embryo is represented by two parts, embryonal axis and two cotyledons. Okay, these cotyledons, they are responsible for reserve food materials. They are filled with some amount of food materials. So, in case of dicotyledonous seed, the site of reserve food material, this is a cotyledon. So, due to presence of food materials inside this cotyledon, these cotyledons are fleshy. These cotyledons are fleshy. That is the site of reserve food materials in case of dicotyledonous seeds. Similarly, embryonal axis, for example, this is embryo in dicotyledonous plants. Okay, this is embryo in dicotyledonous plants. These two, they are cotyledons, and this is embryonal axis. This is a cotyledonary node. Already know the structure of dicotyledonous embryo in previous class. Cotyledonary node. Okay, this cotyledonary node means the point of embryonal axis which is present above the cotyledonary node. This is called epicotyle. This is epicotyle. Similarly, the point of embryonal axis present below the cotyledonary node, this is called hypocotyle. This is hypocotyle. In this way, here epicotyle of the embryo, it terminates into the primary stem or primary shoot, that is called flumule. Similarly, hypocotyle present in the embryo. Hypocotyle. What is hypocotyle? Point of embryonal axis present below the cotyledonary node. This is called hypocotyle. This hypocotyle, it terminates into radicle. That is also considered as primary root system. Similarly, this radicle is also protected and covered by protecting sheath. Such protecting sheath present surrounding the radicle, that is considered as root cap. This is called root cap. In this way, embryo is represented by embryonal axis with epicotyle terminates into plumule, and hypocotyle terminates into radicle and surrounded by root cap. Similarly, two cotyledons are there. These two cotyledons they store food material, but exceptionally very important. In some dicotyledonous seeds, food materials are stored inside the endosperm. For example, in case of castor, okay, scientific name of this castor is ricinus. In case of this castor seeds, food materials are stored inside endosperm. Food materials are present in endosperm, exceptionally in some dicotyledonous seeds like castor. Similarly, some of the seeds, this endosperm is completely absent. They are considered as non-endospermic seeds. Some of the best examples for this non-endospermic seeds, groundnut and pea and bean, etc. In this type of seeds, endosperm is completely absent. Already you know this point. Why endosperm is completely absent in these plants? Because developing embryo completely consume the endosperm, so that endosperm is absent inside the seeds. Seeds without endosperm are called non-endospermic seeds, are also called exalbuminous seeds. Example, groundnut, pea and bean. But similarly, in some members, developing embryo does not consume the endosperm completely. Some amount of endosperm is left out inside the seeds. Such seeds with endosperm, they are called endospermic seeds. And also called albuminous seeds. They are also called albuminous seeds. Okay, some of the examples for this albuminous seeds, that is castor. This is one of the example, castor, which store food material in case of dicotyledonous seeds. At the same time, coconut, Cocos nucifera, coconut. They are best examples for these type of seeds. Students, this is the structure of dicotyledonous seeds. Now coming to structure of monocotyledonous seed. Okay, every monocot seed is represented by seed coat. Okay, here also seed coat is there. Okay, seed coat is there. Within the seed coat, only one cotyledon is there. Only one cotyledon is present. Okay, only one cotyledon it is present in seeds of monocots. Okay, especially in case of Poaceae members. Poaceae members means rice and wheat and also maize. In these members, okay, shield shaped single cotyledon is there. Such shield shaped single cotyledon present in Poaceae members, that is called scutellum. This is called scutellum. Already you know this point in previous class. Shield shaped single cotyledon present in monocot seeds, particularly in grass family, means in Poaceae members like rice, wheat, maize. Such a single shield shaped cotyledon, this is called scutellum. And food materials are generally, they are stored inside the endosperm. What is the site of reserve food materials in dicotyledonous seed? Cotyledons. But what about the site of reserve food materials in case of monocot seed? Endosperm. Okay, but in some monocotyledonous plants, endosperm is absent. Already you know this point. In which type of monocotyledonous plants endosperm is absent? Initially endosperm is not formed. That is orchids. Family Orchidaceae. Orchids. No endosperm. Initially, endosperm is not formed in Orchidaceae members. Similarly, in case of these Poaceae members, okay, seed coat, okay, seed coat and pericarp. Seed coat and pericarp. Seed coat and pericarp completely fused in Poaceae members. Such type of fruits in which the fruit wall means pericarp. The type of fruit in which the fruit wall is completely fused with the seed coat. Such type of fruits are called caryopsis. Such a fruit is the characteristic fruit of family Poaceae members. Such type of fruits are also called grains. They are called grains. What is the importance of grains and what is speciality of grains in Poaceae? Seed coat and pericarp. Pericarp means fruit wall. Seed coat and pericarp completely fused. Such type of fruits are called caryopsis fruits, and generally they are also called grains. They are called grains. Similarly, within the seed coat there is a embryo. So in monocotyledonous seed, embryo is completely situated on the end of the endosperm in a small groove. For example, this is the seed of maize. Okay, this is the seed of maize, or the seed of rice. This is embryo. Okay, this embryo is covered and protected, and this embryo is present in a small groove completely at one end of the seed. Okay, this is endosperm. Okay, major part of the seed in monocot seed, major part of the seed in monocots, that is endosperm, because it's the site of reserve food materials. But what is the major part in dicotyledonous seed? That is cotyledon, because it's the site of the reserve food material. Similarly, this is embryo, and this embryo is also represented by embryonal axis and one shield-shaped cotyledon. This embryonal axis is represented by epicotyle, and epicotyle terminates into the plumule, and this plumule is also represented by some primordial leaf. Already know the structure of monocotyledonous embryo in previous class. Epicotyle, it is this epicotyle terminates into plumule, is also represented by some primordial leaves, and surrounding this primordial leaves there is a protecting gale, that is called, this is coleorhiza, coleoptile. This is coleoptile, and the point of embryonal axis present below the epicotyle, that is considered as hypocotyle. This is called hypocotyle. It terminates into radicle. In this way, seed coat, within the seed coat there is a embryo. Now coming to the most important point, students, major part of the seed in monocotyledonous plants, this is endosperm. Okay, this is endosperm, and in monocot seed, the outer layer of endosperm that separates the embryo from endosperm. Yes, a thin proteinaceous layer, outer membrane of endosperm, proteinaceous outer membrane of endosperm that separates the embryo from the endosperm. Such outer proteinaceous layer, this is outer proteinaceous layer. So the outer layer of endosperm that separates the endosperm from embryo, that is considered as alluron layer. This is called alluron layer. Is made up of protein, because this is chemically made up of protein. But what is the ploidy of alluron layer? Alluron layer is a part of endosperm. But what is the ploidy of endosperm? Triploid. So this is also triploid. So the outer layer of the endosperm, the proteinaceous outer layer of the endosperm that separates the embryo from endosperm, that is considered as alluron layer. Chemically this is made up of proteins, and ploidy of this alluron layer, that is triploid, because this is a part of endosperm.